My name is Ben Chu, and my research focuses on how we can use machine learning to help doctors fight antimicrobial resistance, one of the largest health concerns of our time. Now, these are two pretty big concepts, so let's break them down. First, let's start with the problem. What is antimicrobial resistance? Well, bacteria can enter the bloodstream through cuts, wounds, or medical procedures, leading to life-threatening infections. Currently, we rely on antibiotics to kill those bacteria. The problem is that bacteria are learning how to survive and resist antibiotics, which means the medicines we rely on don't work as well as they used to. Today, when a patient is diagnosed with a bloodstream infection, the doctors will send a sample of their blood to a lab. The lab then has to isolate and grow the bacteria from the blood and test which antibiotics will kill them. This whole process can take anywhere from 3 to 5 days before the antibiotics is administered, which is time a patient with a bloodstream infection may not have. If the wrong antibiotics is given, it also wastes valuable time before the right treatment can begin. Globally, bloodstream infections were responsible for about 2.91 million deaths in 2019. So how does my research try to solve this problem? Well, I wanted to know, could a computer learn to read bacterial DNA and predict which antibiotics will work, giving doctors answers in hours instead of days? Well, let's look at how I did it. What is machine learning? To explain how this works, let's use a cooking analogy. Imagine you're training a new chef. The chef's goal is to recreate grandma's secret recipe, but they don't have the recipe written down. Instead, they're given a list of ingredients and have to experiment, learning through trial and error which combination makes the dish taste just right. This is essentially what machine learning does with bacteria. The computer will take a look at the ingredients, or in this case the genes contained in bacterial DNA, and use this information to determine whether the bacteria will live or die when exposed to a certain antibiotic. Essentially, it learns patterns linking combinations of genes to survival or death. The bacteria's genetic information was collected in a lab and shared with me as data to feed to the computer. Now imagine instead of a few ingredients, you have 15,000 ingredients to go through. No person or even a team of people could reliably find all the patterns across this amount of ingredients. Machine learning is a tool that allows us to process and detect patterns across large amounts of data and genetic information that we could not do on our own. The heat map on the right shows a small sample, the presence or absence of 20 out of 15,000 genes for thousands of bacteria. Now that the computer has learned these patterns, we can give it the genetic information from a bacterium it's never seen before and ask it to predict whether the bacteria will live or die to a specific antibiotic. We then compare its prediction to the real lab results. If the computer's predictions don't match what actually happened, it means the tool isn't ready to be used in a real-world setting yet. To measure how well the computer makes its predictions, we use something called a ROC curve. I won't go into details, but all you need to know is that an AUC score of 1 means a perfect prediction, while 0.5 means the computer is basically guessing at random. Now the results of my research are exciting. Remember, my main question was whether we could teach computers to predict antibiotic resistance directly from bacterial DNA. Out of the 13 antibiotics I tested, 10 achieved scores above 0.95 on the ROC curve, meaning the models could almost perfectly predict resistant bacteria. These results not only confirm this approach works, but also outperform several published studies that use similar methods, showing the strength of our dataset and model design. So why does this matter? If hospitals could use a tool like this, doctors could get quicker predictions about the antibiotics a bacterium is resistant to. That means they could pick the right drug immediately, potentially reducing treatment failures and slowing the spread of dangerous bacteria. Getting bacterial DNA is easier and faster than trying to grow and isolate bacteria from a blood sample. Also, because the model looks across thousands of genes, it might discover new genes involved in resistance, giving us better understanding of how bacteria evolve and helping inform future direction and treatment. Overall, the goal is to reduce deaths from bloodstream infections by speeding up how quickly we can deliver effective treatment. While this project is part of the Lewis Lab, I led the development of this predictor myself, from organizing the data to evaluating and visualizing results. I wrote the code in Python, created the figures as you can see from some of my examples on the left, and ran statistical tests. In a bigger picture, this project connects biology, data science, and medicine, showing how cross-disciplinary approaches can help solve real-world problems. Next, I'm working on a harder problem. Instead of just predicting if a bacteria will live or die, I want to predict the minimum drug concentration needed to kill the bacteria, what we call the MIC value. This gives a much more detailed picture, just like going from hot or cold to reading the exact temperature in a room. So far, I've tested my model on one of the most prevalent bacteria, E. coli, but I will be testing how well this works on other species of bacteria as well. By combining genetics and machine learning, we can move towards a future where doctors treat infections faster, smarter, and more safely. Thank you for watching.